Thank you very much, Jessica, and thank you everyone for being here. So today I'm going to talk to you about a very wide scope project I have, which is to really try to understand what is health and what determines health. But before starting, I would just like to set the stage. I want to set the stage by posing some of the most fundamental questions in biology, but I have to warn you that I will start by answering or offering some definitions from a very non-classical perspective. The reason for that being that I really want to start by giving you the perspective from dynamical systems theory to try to understand or to tackle these questions in biology. So the first question is, what is health? Before answering this question, which I hope we will do by the end of my talk, I just want to introduce this very nice abstract player, which is called the phenotypic space. The phenotypic space is an abstract device, or it's a place where we can arrange attributes of our favorite organism. All of you know that my favorite organisms are cats, of course. So if we would like to place my cat in this phenotypic space, we would have to see, for example, what is the size, so the first phenotype of my cat, what is her age, age maybe not, but what is the length of her fur, what is her weight, but maybe also some more abstract terms like what is the composition of the bacteria in her gut, what is her temperature, what is the composition of her lungs, and so on and so forth. So this kind of way of arranging the instances of biological objects, such as organisms, in such a space, allows us to start really asking questions about how these phenotypic variations, how these differences between us and any other system in the world can be arranged in this space. And we can ask, for example, what is the plasticity of a given attribute? What is the plasticity of, say, my cat during her lifetime? How does she change from when she was born to now that she's a bit older? What is the difference or the diversity between a cat and a snow leopard, maybe there, between a healthy cat and a diseased cat? And how does a system respond to transient perturbations, such as maybe flying to Germany and going to quarantine, but then going back to a healthy state, versus a non-transient response that might lead to a more dramatic and hard to reverse phenotype. Moreover, this kind of way of thinking can also allow us to pose questions about trajectories. So through which region of this n-dimensional space can we move from, say, a healthy region to a diseased region, and through which trajectory can we move back to a healthy region? So I will be working with this phenotypic space concept during my talk, and I would like to pursue you that it is useful to think about very fundamental questions. For example, having this, we could ask what determines the phenotype. So what is the underlying mechanisms? Why are some instances of my biological system here, and why are some there? What causes the fact that maybe some of the biological systems are clustered here and some are clustered there? And sorry, before proceeding, I just want to really thank the library who has helped me to dig out these very amazing pictures from Bauhaus and some others that we will see very soon. Another very interesting question that we can pose is what is the effect of perturbations? So what would be the effect if having different genetic variations or environmental stressors in moving across this phenotypic space? So what would happen if a healthy person, for example, suffers a mutation, will it stay in this confined region, or will it move far away from this space? So it's very natural to think about these questions from a dynamical systems point of view, because in dynamical systems, we ask about the dynamical trajectories of such complex or n-dimensional objects and how these dynamics emerge from underlying interactions. So in other words, something that we would like to be able to pose is, what is the map between the space of possible perturbations, for example, the space of genetic alterations or environmental alterations, onto the space of phenotype? So this is a more abstract representation of what I'm talking about today. So specifically, we would like to find a function that allows us to map, to construct this mapping from the space of perturbations, which we call parameters, to the space of trajectories, which correspond to the phenotypes. In other words, what we would like to do is to uncover, so sorry for going back, by finding or by looking for this F, what we would like to do is to start uncovering disease mechanisms. And so mechanisms through which the system moves from one state in the phase space or in the phenotypic space to the other. And here I want to start by acknowledging the first student I will mention today. So this is Elisa. You will hear about him more in just a bit. But I really like this view of us as modelers trying to predict what is going to happen to a biological system. But to do this prediction, we have to have something in our mind. We have to have a machine that allows us to make this sort of predictions, and these machines are mathematical modeling. So if we have a mathematical model in mind, then this is the dream. The dream of us modelers is, by having a mathematical model, we will be able to observe the system today, given meaning in one region of the phenotypic space, and to be able to predict what is going to happen in the future or in response to in the future, so in delta t units of time, or in the presence of a given perturbation. So now the big question is, how do we construct this function? So until now, I hope I have convinced you that it's really important. So to make this mapping between the space of perturbations and the space of phenotypes, we need this machine. We need this function. And so the next question is, how do we construct this function? How can we construct this mathematical object that will really allow us to make predictions in the way this glass bowl could do? So here again, the key question the key concept are regulatory networks. And for this, I'm very happy that Szabolcs already told you a lot about networks, so you're all very familiar with this concept. And here I just want to very briefly say that in biology specifically, we have networks of regulatory interactions across all levels of organization, and also between and within all levels of organization. For example, we have networks or regulatory networks formed by molecules, for example, proteins that activate each other, or enzymes that activate each other, or transcription factors that activate genes, that in turn activate transcription factors, that in turn activate or inhibit genes. So we have at the molecular level, which might be a bit abstract, I hope not too much, networks of interactions. But we have also at the cellular level, for example, interactions between a bacterial cell versus a cell of our body, a cell of our body in one part of our tissue versus a cell of our body in another part of our tissue. Again, they form regulatory interactions because there is talking to each other. We have also regulatory interactions at the tissue level, at the organism level, and of course we can scale up to ecological level modeling in trying to understand community assembly and other really interesting questions. So these regulatory networks is really the meat. This is maybe a Spanish expression, but like the essence of what we will use to construct our mathematical models. We will construct mechanistic mathematical models that represent these regulatory interactions at different levels of organization. So basically what we do when we construct models is to take such a network of such an interaction network, such a regulatory network, in this case, and you will see a lot of this today, this is a bacterial cell, this is an epithelial cell. Keep this in your mind because we will see plenty of those, and those are immune cells such as neutrophils and macrophages. So we translate this representation, this graphical representation, in a formal way, in a formal representation using dynamical systems. And the advantage of that is that we can borrow tools which have been very nicely developed in engineering and other more quantitative sciences to really squeeze out all the information, all the emergent properties from those interactions. So here the key thing I want to say is that we construct mechanistic models because they represent the mechanisms of interactions that underlie a given property, such as health. So now that we know, at least we have the intuition, hopefully, of how we construct these models, the question is again, what is it that we want to predict? Which kind of predictions we want to do now that we know how to construct our model? Well, again, the predictions that we want to do, and maybe this might sound a simple answer, but it has very, very large or wide implications. We would like to be able to predict the response of our biological system to different types of perturbations. So again, framing it back to a dynamical systems terminology, what we would like to do is to map the space of perturbations to the space of trajectories of phenotypes. So basically, so one of the key concepts here is that we see biological systems with these regulatory interactions as signal processing devices, which basically take decisions, for example, phenotypic decisions, health versus disease decisions, as an input-output relation or a decision-making device. So with this, we are now ready to start seeing health as an emergent property. So basically this confinement or movement in this phenotypic space as a property that emerges from the regulatory interactions that we can encompass in our function f, which represents the regulatory players. So which is the kind of questions that we can tackle with these kinds of approaches? So if we use our mathematical model representing these regulatory interactions, we can ask many things. For example, we can ask what are the mechanisms of change that make us move from state A, maybe a healthy state in the region of the phenotypic space, to state B, a unhealthy region in the phenotypic space. We can also ask what is the combination of perturbations that can be tolerated by the system that will not make the system move from state A to state B. We can also ask if a trajectory, can we predict that a trajectory is going to go from a state A to a state B by something that we call early warning signals? And are there ways to optimally prevent these transitions? And moreover, how can we reverse these trajectories? What is the best way to move back from this state to this other state? In other words, are there optimal intervention strategies that minimize negative side effects and minimize costs? So in other words, what we really want to understand using our mathematical models is how is the balance between homeostasis, between robustness and plasticity, which is responsible for the maintenance of homeostasis, meaning that the system is able to dynamically respond, but without moving away, too far away from its setting point, through the regulatory interactions. So we want to understand what maintains and also what tilts this balance. So a typical pipeline that we use in mathematical modeling, specifically in the type of mathematical modeling that we and others do, is to always start by the data. So although I have used the word toy a lot, but in fact, we really want to use models that can accurately represent the data and that can also inform us about specific instances of the empirical world. So we normally start by really gathering plenty of experimental and clinical data that we have at hand, and then we integrate it and analyze it by constructing our mathematical model. So this funnel represents basically this process of really bringing back putting together the essential observations that can give rise to these key mechanisms that could maybe account for a given observation. So the aim here, the output of this part of our pipeline, is that we would like to construct minimal models that represent plausible mechanisms that can account for pathogenic mechanisms, for pathogenic observations. So once we have our mathematical model, we then go to model analysis. So we start hitting our system and assessing which kind of hits or which kind of perturbations will account for an abrupt movement from a health to disease transition. So here, what we want to get from this part of our analysis is to be able to predict what will be the effect of risk factors such as genetic alterations or environmental perturbations, and also to be able to say something about the possibility of early detection through early warning signals. Every time, so here, of course, because those are predictions, we have to go back to the empirical world and to see if our predictions actually hold. So normally, although I present this here in a sort of, my narrative is sort of linear, but we're always going back and forth. So the models I will show you today are maybe the version n with n much bigger than 1 because we have gone through this cycle quite many times. So once we have done this, so basically we have identified possible disease mechanisms, then the next step is to basically plug in our model to our favorite algorithm, which will allow us to optimize a given treatment. For example, we can use something like model predictive control and other nice computational tools that you might be familiar with because they have been vastly explored to solve this very nice puzzle of how to stabilize an inverted pendulum. So it might sound quite abstract, but it's a very dynamical systems problem. So what is the best control mechanisms to stabilize this unstable state? This is the kind of tools that we can borrow to analyze biological systems. And why does it matter? So it's very lucky for me that we have been already 2 months here. So I've been able to talk about this with many of you. And those are some of the notes that I literally had on my desk of some conversations that we have had here. So for example, during the first, a couple of weeks ago, someone mentioned this big problem that we are drowning in data and that in biology, we have to make sense out of this data. So constructing this type of mathematical models is our answer to this question of how to avoid drowning in data. When talking about this, and this is something with Sabina, we have talked from the very first day. I was very impressed by her very sharp way of understanding the key problem in this data integration is this standardization. This kind of modeling also points the finger towards the urgent need of creating standardized databases that allow us to integrate them in a formal and coherent way. So it's a way to allow us thinking about this kind of problems in a more rigorous manner. With this kind of models, we can also try to look for the simplest possible explanation for a given observation. We can also try to identify what are the limits of a given system, so a system that can account for our observation. And finally, we can construct a toy through which we can conduct in silico experiments. So a representation, a formal representation. So with this, I come to my first act, and I will move to my, I have to say it, to my comfort zone. So this is what I have been working most during the last 10 years or so, is in modeling epithelial tissue diseases. Specifically, I modeled these systems trying to understand what are the mechanisms of health to disease transition. And why are epithelial tissues interesting? Well, they are really interesting because they are the very basic tissue of any animal, including sponges, which are non-vertebrates, which basically mark the limit between us and the environment. And therefore, these tissues have an extraordinarily important role in really maintaining this balance between robustness and plasticity. Because on the one hand, we and sponges and any other animal you can think of have to be able to interact with the environment, but at the same time, we have to be able to maintain our identity. So this means that this question about robustness versus plasticity is particularly relevant when we talk about epithelial tissues, and also talking the last days with Jessica, something that can also be posed in this framework are evolutionary implications, or how different epithelial tissues have changed across different species and so on and so forth. Today I will not talk about this, but it's something very nice to think about. So in our bodies, epithelial tissues, examples of epithelial tissues include the epidermis, which is the uppermost layer of the skin. I will talk about this today, just in a second. The airway epithelium, which if you have talked with Rachel, you have heard about it already, and if not, we all look forward to her talk as well. And there are other types of epithelial tissues I will not talk about today, but which we have also studied in my lab, such as gut, cervix, mammary epithelial tissues, and also hepatocytes, which form the liver. Today I will focus on epidermis and airway epithelium. So what are these tissues? So they are formed by specialized cells. So this sort of Bart Simpson's type of cartoon represents the epithelial cell. You can already see here that they have this distinctive feature of polarity. So we have a very clear distinction between what is outside and what is inside our system. And so we not only see that with this apical distinction in the form or in the phenotype of the cell, but also we have this clear barrier. So this sort of hands, they are not hands, they are proteins called, for example, desmosomes or tight junctions that really held these cells together and avoid the penetration of environmental factors or environmental triggers such as bacteria, and also avoid, for example, that we get dehydrated the minute that we are born. So it really regulates the exchange with the environment, and therefore it's really crucial to maintain this balance between responsiveness, so between what is outside us and what is inside us. So when things go well, which fortunately most of the times it's the case, then we have this regulated exchange with the environment and a protection against aggressors. So we have also heard about from Britt a lot about microbiota, and this also plays a very important role, for example, in compartmentalizing the bacterial source that we have in our body. So it's, for example, common sense, many people agree on that it is sort of allowed, let's say, to have plenty of bacteria on this side, but this side should be mostly sterile. Or much more sterile than this side. So this compartmentalization is very, very important for us to maintain health. So when things go well, we have something that looks like this in a cartoonish way, But things can go bad in very many different types of ways. For example, maybe we could have an overload of pathogens leading to infection. So we can lose this ability of maintain them out of our system. We can also mount an exacerbated immune response leading, for example, to inflammation, which can be seen, for example, as an increased concentration in cytokines in our system, which is a very, very big problem, for example, in COVID patients, among many, many other diseases. And we can even go further, and there are diseases where we have a phenotypic alteration, where this sort of shape is lost completely. So we lose the polarity, we lose the junctions between the cells, and moreover, these kind of cells could be able to move, so they become motile, and those are the ones responsible for metastatic cancer progression in carcinomas. So we really want to shine light on these health to disease transitions with mathematical models, again, by assessing pathogenic mechanisms, by assessing effects of risk factors, by looking for early warning signals, and by looking for optimal treatments. And for that, I will start with a first example, so skin and its diseases. So this is historically the epithelial tissue I've been working on for a very long time. I did my PhD on this, and still this morning I had a conversation with my then supervisor because we continue working on this system. So it's not the simplest one, but anyway, we have been looking at it for a very long time. So we always start this kind of project by sketching out the regulatory interactions. And here I just want to walk you very quickly through this. So here we have again this environmental pathogen. In this case, it could be Staphylococcus aureus, which is a bacteria that all of us have in our skin. And this bacteria, under some circumstances, can lead to the abrupt activation of immune responses. So the epithelial tissue responds to this bacterial load, sometimes, not always. And in fact, when it does it in a very on and off type of manner. So this is important, so keep it in mind. So either you have a rash or you don't have a rash. So there are no intermediate points. And when this happens, then two processes occur. So first, we have these immune responses, for example, secretion of antimicrobial peptide, which is very nice subset of substances. So they are like antibiotics, but naturally produced. So it's really, really nice to study antimicrobial peptides. So we have a release from epithelial cells of antimicrobial peptides, by epithelial cells, which can then kill and counteract the pathogen. But at the same time, when this happens, everything comes with a cost, as all of us know. So we also have a weakening of the barrier, so a weakening of these junctions between the cells, basically making or allowing the pathogens to move into our system. So basically corresponding to a positive feedback loop. So we have this trade-off between negative feedback, so pathogen comes, we eliminate pathogen, but at the same time, pathogen comes, the tissue will be weakened. So our hypothesis back then, and it still is that it's only through the balance between these positive and negative feedback mechanisms that this that this balance between robustness and plasticity can be achieved. So that when we move or that when we disturb this rate, so these feedback strengths, that we can move abruptly from a health to disease region in the phenotypic space. To answer this question, we construct a mathematical model. So in this case, I told you that this switching module is very important. We represent it in this way. I will not go through the equations today because of the sake of time. But basically, for the mathematicians here, this gives rise to a special type of dynamical systems called piecewise smooth dynamical systems. So I just want to say very briefly that those are, this is a type of dynamical system that is not that common in biological biomathematics, in biological modeling, and therefore this is actually good news because there are also many challenges and open questions in terms of formal tools development and proving theorems and these kind of things that you've heard about a lot in Sabol's talk for networks. So we formalize these interactions into a system of equations where we have this switching function, and also we have this a slow variable that acts as a capacitor, which will become important later on as well. So then when now that we have constructed this toy, we can start asking our questions, right? And what is the question that we want to ask? Well, we want to understand what will be the effect of perturbations on the phenotype. And which perturbations we choose. So in this case, we knew from before what the most prominent genetic alterations that could lead to a loss of health in the skin are. So basically, they correspond, in fact, to weakened or to alterations in the feedback strength. So one of them corresponds to a weakened immune response, to a low ability of secretion of antimicrobial peptides, and the other one corresponds to a basally more permeable barrier given by a lack of expression of these tight junctions between the cells. So what happens when we continuously tune or move these two parameters together, so these two genotypes together, so basically explore all the possible consequences of different instances of changes in two parameters. So what we can see here is that the phenotypic space, so the space of how the skin would look like, will be divided into 4 clear-cut regions. One of them is a healthy region, is also the largest one, which is good news because health is a robust property of the biological systems. And then we have 2 other regions, which I will not go into details today, but we term them susceptible regions. So it's the disease phenotype is not fully blown out. And then we have this red region, which is the severe phenotype, which is like a very severe skin disease. This is really what we want to avoid. So this is like the first type of nice result that we get by doing this kind of modeling, that we can segregate the phenotypic space into clear-cut region. And actually, this clear-cut mathematically corresponds to these curves, which are called bifurcation curves, which in this case, in particular, because it's a special type of dynamical system, it's also a special type of bifurcation. They are called boundary equilibrium bifurcations. We can then ask, okay, what will be the effect of treatments on these phenotypes? So again, we can pose this question in terms of looking for bifurcations. But now, instead of having risk factors, genetic risk factors as perturbations, we will use the treatments, and we will systematically assess what is the effect of all possible combinations of treatments, in this case antibiotics and corticosteroids. Corticosteroids dampen the inflammation. So how can we achieve an abrupt transition from a disease phenotype to a healthy one? And we can answer that in a systematic way, again, by looking at this boundary equilibrium bifurcation curve. So these regions in the space that, again, give a very sharp and clear cut into these different domains. And they correspond to all the optimal combinations, all the optimal solutions of combinations of antibiotics and corticosteroid treatments that can drive us to this region of the phenotypic space. Finally, well, we can also ask about early warning signals. So if we're in the gray region, we really want to avoid entering the blue and avoid even more entering the red one. Given that it's a qualitative transition, are there markers that tell us when this catastrophe will happen? We know from dynamical systems theory that the answer, at least in classical system, is yes. Currently, we're working in looking if in this kind of systems, we also have these early warning signals, and this sharp rise here indicates you that the answer, again, might be yes, which is super cool. And then we can also use our model to devise optimal treatment strategies, in this case with model predictive control, by again designing the way to move a patient from this phenotype away and towards this healthy looking phenotype. So we can use model to say what would be the best strain of corticosteroids and emollients to move from this severe state to a healthy looking, this is why it's blue, not red, state. We can do something similar when modeling infection of the upper airway epithelium, in this case by Streptococcus pneumonia. I just want to show you here, so this is a different type of epithelium. It's in the upper airway, meaning in the nose. Here the bacteria is again a different one, it's Streptococcus pneumonia, it's not Staphylococcus aureus. But again, the control structure, I hope that you already noticed that it's very similar to our previous one. So again, we have environmental stressors, we have barrier integrity, barrier function, we have immune responses, we have again the balance between positive and negative feedback, and again we have these switching functions that mediate the turning on and off of these responses. So with this, now I don't show you the equations, but it's similar to the last ones. So with the equations, we can then really ask the equations to reproduce also in a quantitative way all the available experimental data sets. And this might sound trivial to some of you, but actually I spent a good part, most of the part of my first postdoc just doing this. It's not trivial at all because, again, this question of standardization of the data sets, like measuring how many milliliters of bronchoalveolar lavage a mouse has, and if it is comparable to this experimental condition in a Petri dish, yes or no. So it was very, very nice to be able, with our model, to reproduce these empirical observations. So basically to see these observations as an emergent property of the regulatory interactions of this wiring, of this regulatory network. With this, we can then ask again how robust the system is. Here again, we would expect that the system is able to withstand the vast majority of perturbations because, again, most of us are healthy, fortunately, and indeed this is what we see. So if we perturb the system in random ways in all possible directions in the perturbation space or parameter space, we see that a vast majority, around 80%, will remain healthy, and some of them will be severely ill. And then we can also ask, so we would like to ask, so what is responsible for this dramatic change between a healthy response and a totally unhealthy one? For this, we can do, for example, something called parameter sensitivity analysis, which is a computational tool that can be applied to this kind of mathematical models that allow us to identify vulnerabilities in the system that can potentially lead to abrupt phenotypic transition. So this is very important to narrow down the space of all the possible or the necessary experimental conditions that we have to test in the lab. So with this, from all this n much bigger than 1 possible responsible facts, we can, or responsible mechanisms that could lead to this change from health to disease, we can really point at these 3 that are most prominently or much more likely to lead to this dramatic transition, and then go back to the lab and do these 3 experiments, but maybe we don't have to do all these other n bigger than 1. Something similar can be done with tuberculosis. So this is the work of Elisa, who is just about to finish, well, actually, finished his manuscript, he finished his thesis already. So again, he spent a big part of his master's assembling a standardized database, which he then used to construct a minimal mathematical model to represent the key host pathogen interactions that occur in granulomas. So in this case, it's macrophages and the Mycobacterium tuberculosis. And something important here, which are the small symbols here, which are very hard to see, but I blew them out here again. We have, again, this switching mechanism and this capacitor giving history-dependent changes in the kinetic rates that occur through modifications of the adaptive immune response that leads, for example, to a secretion of hormones and neurotransmitters that, by the way, can account for this changes in mood that already Susan Sontag described in one of her books. So with this, we can then also ask, okay, what we can, again, fit data and ask if this model is able to robustly reproduce all the available empirical data. The answer is yes. And finally, we can ask interesting questions, such as if the system or these host pathogen interactions progress, or as these host pathogen interactions progress from early to a middle to a late stage of the disease, does bacterial clearance, so a healthy outcome, versus uncontrolled bacterial infection become more or less likely. So we can use the model to really calculate the probabilities and also to assess the mechanisms of progression from one stage to the other. And finally, this is very briefly just the work of Rebecca, my other very bright master student. And she worked not with experimental, but with clinical data of SARS-CoV-2 patients. She assembled a large database with longitudinal data and then constructed a mathematical model that represents the underlying mechanisms that could underlie these different dynamics. Just this morning, so literally out of the oven, because my computer was very hot, we were able to fit this model to this data. And what she is doing now is to really try to identify those parameters that segregate these cohorts into non-severe and severe responders, to really identify what could be the potential causes that change this phenotype in such a dramatic way. So with this, I finish talking about epithelial tissues. I just want to quickly sum up about the common network properties that we have encountered in these 4 examples we have briefly walked through. So one of them is that all of them share key regulatory players. So we have 4 main characters in these stories when talking about epithelial tissues. We have, I will call them microbial components. We have also immune cells or immune mechanisms, such as antimicrobial peptides, but also others. And we have the barrier function. And they are connected in non-trivial ways by negative and positive feedback interactions that look more or less like this. So we have different positive and negative feedback mechanisms that together balance out to allow us a healthy and homeostatic response to perturbations. And we have also encountered these 2 nice network motifs, so these abrupt switching functions that give rise to this all or nothing type of responses, and also this sort of capacitor which gives rise to history-dependent phenotypic decisions, as in the case of tuberculosis. So in a very, very brief Act 2, I will just try to zoom in and zoom out of my comfort zone to see if this type of modeling can also be applied to other levels of organization. And for that, I will present only this slide, so don't worry, it won't take too long. So far I've been talking a lot about epithelial tissues, specifically about skin and about airway epithelial tissues, both here and also in the nose. I've told you that we have also worked on other epithelial tissues, although I haven't talked about this today. But after working like one epithelial tissue after the other, we sort of wanted to ask if this kind of model with this kind of network motifs would be able to also explain other type of empirical observations. So for that, we started looking at cellular level development of a fungus called Neurospora crassa, and it's a very interesting system. So it's a organism which is undifferentiated under non-oxidative conditions. So if you don't take it out to the atmosphere with air, it will not do anything. But as soon as it has contact with a high oxygen level, it starts to go through a process of development, of differentiation. And interestingly, this process of differentiation occurs as a response to this environmental trigger, of this environmental perturbation, and is mediated again by barriers in this fungus. So we wanted to ask if we were able to reproduce these empirical observations, again, this dynamical trajectory in the phenotypic space, this time not from health to disease and back, but rather from an undifferentiated to a differentiated state in response to a perturbation, which in this case is oxygen. And here again, so this is very small, I apologize for that. But again, we constructed a mathematical model where again we have these switching functions and this capacitor motif. And the answer is yes, we can reproduce this in a nice way. We have also looked at some population level dynamics. This is, we have talked over dinner with some of you about this as well. So this is to model what happens in a bioreactor. So here the question is really nice because it's like how to construct mathematical models to optimize brewing beer and other alcoholic beverages. So this is the work of another very bright student of mine, Juan Manuel Gutiérrez García, in collaboration with Juan Carlos González in Morelia, back in Mexico. So they, Juan Manuel constructed a wide set of mathematical models to reproduce the data observed in bioreactors to answer the question, what would be the optimal conditions to get the maximal yield out of a fermentation process. And the largest scale I've worked on is in the establishment of stable ecological communities. So some of you might know that Mexico is one of the most biodiverse countries in the world, and therefore we have very, very nice places where, very nice and also unique places where different interactions between native species happen. And it's of great interest to study how these interactions occur and, crucially, which perturbations to avoid so that these ecological community interactions are not destroyed. So for that, we worked on two different projects. So one is a plant-animal model of a closed model of plants and animals, and there we looked at coevolutionary implications, or what are the conditions of coexistence between these two species versus those of local extinction. And something similar, we did something similar for a tree community in Mexico City. It's a very special place because it's very young in terms of, so it's volcanic eruption happened only 2,000 years ago, meaning that it's a new place. In ecological terms, it's quite new. So we have a still establishing ecological community of trees here, but we have the big threat of eucalyptus that people think that will destroy this assembly of this native species community. So we constructed, with all the data that we had, a mathematical model, and again we see this clear-cut separation in this space from a healthy, or we call healthy, coexistent of this native species versus the complete dominance of this nasty eucalyptus species that basically releases a lot of toxins in the soil. So with this, we would finish this talk with a little bit of alchemy. So we have been looking at many different models across different scales, mostly epithelial tissues, but I've talked a bit also about yeast modeling, in development, and even about tree community assembly, and so on. So one key question for us as biomathematicians is what are the key structural features of all of the systems? So what do all of these systems have in common? Can we say something about that? And this is actually the big part of the project I proposed for Vico. So this is very much work in progress, but I just want to show you one slide regarding this. So in mathematical terms, what we want to look here is at something called normal forms. So normal forms are the simplest possible mathematical expressions that give rise to a given bifurcation. And it's super useful to have this kind of mathematical objects because it means that through analyzing these mathematical objects, which are as simple as it gets, we can say something about more complicated objects, but which have some addendums that are not structurally necessary for the qualitative observations. So our aim here was here to is still to construct the simplest possible yet biologically plausible network motif that gives rise to these slow yet abrupt and history-dependent phenotypic transitions. And maybe you have already guessed what are some of the key elements that this object, this mathematical object, has to contain. So one of them is, we need a switching function, we need a switching core, which is this pink one. Actually, with Marco Tulio Angulo, a colleague of mine in Mexico, we call this the egg project because it has 3 layers. So the yolk is this switching motif. So we have this that you have seen many times, to this abrupt on and off phenotypic decision core, which is then covered by its yolk, like the white part of the egg, which is a slower layer, which has this balancing between positive and negative feedback strength, which modulates and is modulated by this bistable core. And finally, we have this shell, which we call the ultra-slow layer, which basically corresponds to this capacitor, which further modulates the strength of this feedback strength in a much, much lower time scale. And so far, we have analyzed these 2 layers, so we still have to plug in the ultra-slow one. This is something that we're doing now together with Pablo Aguirre, who is a really extremely bright person, expert in dynamical systems from Chile. And we are about to test some of our model predictions with the simplest possible system where we have observed something like this, which is again in yeast, but this time not in neurospora, but in Saccharomyces cerevisiae in the lab of Dr. Alejandro Coleman Lerner in Universidad de Buenos Aires. So what we see here again is that when we modulate the feedback strengths, we can again get this nice segregation into 4 distinctive regions in the phenotypic space, and we can really explore how the attributes of the switch and exhaustively tuning these parameter strengths can modulate or change how this phenotypic space segregates. A nice thing about working with a simplest possible model, and this is why we reached out to Pablo Aguirre, who is a real mathematician, is that we can actually say analytical things. So not only through simulations, so through numerics. So most of the results I showed today, we have to program stuff and simulate, but if we have a model that is as simple as possible, we can actually say with total confidence what this object can do. And we can do this by proving theorems. I don't do that. This is what Pablo Aguirre does, but this is quite nice. So we can really show that we are really about to show that this is the biologically plausible, simplest possible normal form responsible for this phenotypic segregation as a function of perturbations in parameter space. So with this, I come to an end. I just wanted this is actually the slide I used for my interview for Vico. And normally, we never do what we promise. But I have to confess that. But this time, actually, yeah, it's surprisingly, I think I am actually going into this direction. So it's going sort of well. So basically, we have been using mathematical models to really try to shine light on different mechanisms of health to disease transitions across different levels of organization. So again, my comfort zone is in epithelial tissues. I think it's a very nice scale that also allows us to ask different types of questions, also evolutionary, developmental, of course, physiological, and so on and so forth. But indeed, branching out, zooming out and in, away from this comfort zone has allowed me to identify some structural properties of these wiring diagrams of these regulatory networks that could also be used to explain other types of phenotypic transitions at different levels of organization. So with this, I want to thank you, and I can't believe it's just on time. Thank you very much, Jessica, and thank you.